cell communication. So, what we're going to do is do a brief sketch of a structure that you guys have learned a ton about. And that structure is the plasma membrane. And so I'm going to use some colors to differentiate between the different structures of the plasma membrane. And we'll go back and label these here in just a little bit. And so the first thing you want to do is make a protein that looks like the pink one or the magenta one over here, and make a protein that looks like the orange one. And I am drawing circles to represent the phosphate heads of the phospholipids that I am now going back through and adding the fatty acid tails to. So keep in mind the different names. This is called the phospholipid bilayer. In this case, it's the plasma membrane. We have to be careful about that, though, because we have learned that cells have lots of membranes. They have a whole endomembrane system, and all membranes, whether it's the plasma membrane or the mitochondrial membranes, outer or inner, or the chloroplast, outer or inner membrane, or the thylakoid membranes of the chloroplast, or the nuclear envelope, which also has an outer or an inner membrane, and the smooth and rough ER have membranes. The Golgi apparatus has a membrane. One of the only things in the cell that's a cell organelle that doesn't is the ribosomes. And so we have to be sure that we are figuring out what kind of membrane we're looking at. And since we were looking at cell communication, we're going to say we're looking at a plasma membrane. And again, another name, fluid mosaic model, because it's constantly moving, it's not in stasis, and it's a mosaic because it has not only phospholipids, it also has proteins. As we just looked a second ago with cell-to-cell recognition, it has carbs, it has those cholesterol molecules. Again, the latter two not shown. But we're going to get a little more specific than just calling this the plasma membrane. And we're going to call this a muscle cell plasma membrane. So on one side of this membrane, I'm going to label this as the bloodstream. So this would be, the bloodstream would be considered the extracellular fluid. On the flip side of that, we're going to label this down here the cytoplasm. And the cytoplasm would be composed mostly of the cytosol, which is composed mostly of water. Okay? So here's what happened more than likely, or at least I hope you've done this at some point today. Probably pretty recently, you had some lunch, I would hope. And when you had some lunch, you probably ate some carbs. Now, whether you sat down and ate a bunch of sugar, or whether you had a sandwich, or something that had more starch or fiber or what have you in there, what ended up happening is your blood glucose levels increased. And so it gets that blood glucose out of balance. And when that happens, your body has to respond. So going back to the characteristics of living things that we did in the very beginning, we have response and regulation, and they're often coupled because a cell usually responds so that it can regulate the external and internal environment. Now, in this particular example, it needs some help. And so most of you know that whenever you eat sugar and your blood glucose level goes up, your pancreas cell makes insulin. And we'll draw this little yellow thing here, and we'll say that that's insulin. Insulin is a protein hormone, and it's worth noting that because protein synthesis had to occur for it to be produced. And insulin is going to require an extracellular receptor. And so that's what this guy right here is. This is a receptor protein that receives chemical signals. And so insulin is specific for that receptor protein. So what it's going to do is it's going to come in contact with this receptor protein. And also keep in mind, I'm oversimplifying this. We're going to look at how the insulin is produced in Unit 6 when we get to protein synthesis. We're also going to look in that same unit with biotechnology, how we use bacterial cells and recombinant DNA and plasmids to produce insulin for our diabetics. So we're going to get into the biotechnology side as well. So when this happens, we have the first step of cell communication, which is reception, okay? And another thing I want you to point out is that the insulin would also be called a ligand because that is our generic term for a hormone or a signaling molecule. So reception is the first step. Now, again, I'm going to oversimplify this. We're going to look at some of the details a little bit later on. But what this does is this results in a message being passed from the receptor to these things that we'll call transport proteins. And what that message says is it says, hey, our blood glucose is super high. You need to let glucose in. And what happens as a result of that is glucose enters the cell. And so regulation of blood glucose levels has to do with both cellular transport, but also cell communication. And just as a reminder, as a recap, we are in an animal cell, we are in a muscle cell. And so once inside the cell, there are a couple different pathways that glucose can take. So if the cell is currently meeting its energy requirements and doesn't need that to break that glucose down through glycolysis so that the process of aerobic respiration can occur, then that energy is stored in the form of a storage polysaccharide in animals called glycogen. But if the cell's energy requirements are not being met, then it can take a different path. And that glucose can be split in the cytoplasm. And again, that's called glycolysis. And then it can go into the mitochondria once it's split and goes through that process to ultimately produce energy in the form of ATP.